So UP421, let me tell you what it is. So this is what we did is we gene modified islets from the pancreas of someone who had recently died, just had died. And so we knocked two genes out and we knocked two genes in. And so then these cells were transplanted in the muscle of a patient. And what is so amazing about this is this patient has gotten someone else's cells and no immunosuppression, and the cells are alive, functioning, and evading the immune system 14 months later. So what should happen, and what would happen normally, is they will be rejected within a matter of a couple of days. Our scientists believe that once we got into 28 days, we were in the clear, like nothing else would get to us. It turned out, right, that that's been true, right? And we're now out 14 months, and the patient continues to make his own insulin, and these cells evade the immune system. And just to say that one more thing, this is actually more complicated than it will be in most situations, because not only did we have to overcome allogeneic rejection, right, putting someone else's cells into his body, but he already had a pre-existing immune response to these cells. He has type 1 diabetes. And so we were able to overcome that as well. And I think it gives a lot of people a lot of hope that we are at a period where we can realistically believe that there is a cure on the horizon for type 1 diabetes. And to your point, this finding should extend to all kinds of other cell types and broadly for the cell therapy field. One of the really spectacular things in technology evolution is when a new technology moves from being kind of the product to when it just becomes kind of a reagent or an input into a broader product. So what I mean by that here is CRISPR gene editing. So CRISPR gene editing has been thought of as this way to do all what to maybe make new therapeutics to go after certain gene disorders that are very targeted. And what we have here is the ability to go after a couple of genes, to knock two out and knock one in a very precise way into an exact location in the genome, and create a broadly available therapeutic, hopefully for patients with, most patients with this disease. And so what I love about it is the next era of precision medicine is we get this opportunity to go after diseases that impact millions of people. And it's really because we've taken a new precise technology, which is, CRISPR gene editing, and been able to apply it in a way that it's a part of the story, but not the only part of the story, which is when technologies are truly, truly beginning to have their biggest impact generally. And I'll say, this is a combination of four really important areas of scientific advance. One is stem cell biology. Two is CRISPR gene editing. Three is transplant immunology and really understanding the immune system. And then four will be manufacturing sciences. We have to get them all right to really make this into something that we hope it can be for patients.